Dr. Dennis Schamhart has a Ph.D. in biology and was at the Department of Molecular Biology at the University of Utrecht in the Netherlands. His research is in the application of electromagnetic fields as a therapeutic technique with special research at Rutgers University and the Dutch Cancer Foundation. And now to discuss his work in photon emission in normal cells versus tumor cells, plus his research in hypothermia, I give you Dr. Dennis Schamhart.
Okay, good morning, ladies and gentlemen. Now projected, you see the title of my presentation for today. But before I start explaining this title, I would like to thank the World Research Foundation for their kind invitation and the opportunity to present my work for this select audience. Reading the abstracts of this symposium, we probably all noticed that several of the contributors will report their results and ideas on subjects not commonly accepted by the Western scientific community. These speakers will introduce to you various diagnostic techniques and therapies which are not taught within school medicine. Paradoxically, various of these diagnostic techniques and therapies do know a long, sometimes very long, history in medical treatment of humankind. So, for example, acupuncture is already known for more than 4,000 years. In addition, it seems, however, worthwhile to notice to you that the presented techniques and therapies are by no means the only ones currently in practice within the field of the so-called holistic medicine. Indeed, the really overwhelming variety of diagnostic techniques and therapies within this field seems to be one of its main characteristics. In a recent survey, Utrecht, at my laboratory, collected and described more than 200 of such diagnostic techniques and therapies. I mention this to you because it seems likely that exactly this impressive number and variety of techniques and therapies, all claiming their own particular approach to the human body, hamper research and consequently the process of acceptance. In the eyes of an outside observer, this field is highly chaotic and no coherence can be found in the still fragmentary research. Research in this field hardly knows a tradition or an accepted methodology. In the light of these facts, it seems to be clear that there is an urgent need for a unifying concept, structuring and preferentially embracing as many as possible therapies. On the other hand, such a concept needs to leave the therapy-specific characteristics intact. Most important, this concept should have a solid biological background, initially at least at the theoretical level. Such biological concept should open the possibility to study the effectiveness and working principle of the various therapies according to the methodology and standards of natural science. In my opinion, such a concept may provide the link between physics and medicine, which happens to be the subject of this Congress. The link must be at a structural biological level. It's for this reason I decided to introduce to you the current biological research devoted to this subject, explaining the title of this presentation, Holistic Medicine. Is there a biological rationale? The next slide, shows the people participating in this project at the University of Utrecht down in the Netherlands. As they are Roland van Wijk, Wilbert Lindemans, Vet Wiegand, and Wiel Geilenkirchen. In an initial approach towards the development of such a concept, we analyzed the various types of holistic diagnostic techniques and therapies and looked for common denominators. Without going into detail of all those techniques and therapies, this study revealed that a large number of therapies stresses the biological organism, or more specific, the human body as a whole, interacting with its environment. A living being, so said, has the inherent ability to regulate or adapt its activities, its functions of life, and physiological processes in an optimal manner according to the actual environmental conditions. This part of the view treats the interaction between the organism and its environment. In addition, mutual interactions between the various parts of the organism, the cells, the organs, so on, and between the surrounding fluid are considered. This view, graphically represented in this slide, bears a great analogy with the view of the French biologist Claude Bernard, who already in 1859 formulated the existence of an external environment, milieu extérieur, and an internal environment, milieu interne. In this scheme, the arrows indicate the interactions between the various entities. The functioning of this extremely complex pattern of interactions between the various parts of the body and between the body and its environment, expressed as a continuous process directed towards a state of equilibrium, is seen as the central topic in a large number of holistic therapies. Illness, in this view, is considered as a malfunction of these interactions, which may be corrected by the various techniques used in this field. Interestingly, expressed in these commonly accepted biological terms, holistic medicine does not appear so out of reach as may be concluded by a first glance. Essentially, we are dealing with, although extremely complex, cybernetic control systems, like feedback control circuits. The techniques used in the various therapies seems to interfere with these circuits. We're now left with the question, can we find an array at which these interactions are expressed at the material level? For it's at this material level we will be able to collect quantitative data about these interactions according to the standards set by the natural sciences. In this respect, me too, I have to go to the work of Piesinger, and it's turned out to be already the third time his name is mentioned here at this Congress. It shows how important we in Europe think that his ideas were. Furthermore, last night at a dinner I was honored for presenting you with at least a 60-minute presentation, so I will go on in the Piesinger model too. Oh, more seriously, there is another reason. My point of view will be a little bit different from the point of view presented by Dr. Beck and Dr. Stadtmacher. So, the work of Piesinger and his co-workers, they were studying 2 to 4 decades ago in Vienna the morphology, histology, and biochemistry of the loose connective tissue of mammalians. In his opinion, the loose connective tissue must not be considered as a mere rigid supporting tissue without any additional specific functions, but on the contrary, as a tissue performing, regulating, and coordinating functions in the body. The loose connective tissue interweaves almost the complete body and seems to function as a dynamic barrier and communication channel between the specific organ cells on the one hand and the blood capillaries and the autonomic nervous system on the other hand. The next slide shows, without going into great detail, this so-called System der Grundregulation, system of ground regulation. This system surrounds the organs, here depicted as organ B, and somewhere over there is organ A. And it consists of 3 major components. This slide shows you the first component. And here we recognize the humeral component. It's spelled in Dutch, but I think you can understand it. This humeral component with blood capillaries, which are depicted as A, and lymphatic tissue, B, are the anatomic elements. Furthermore, it consists of the extracellular fluid, composed of materials derived from the blood plasma and organ cells, all in process of diffusing back and forth between the capillaries and the organ cells. Furthermore, we find here the amorphous ground substance composed mainly of mucoplasma. Fibroblasts play a crucial role in the regulation of this extracellular fluid, synthesizing, for example, hyaluronic acid and growth hormones. In addition, they seem to regulate the oxygen, acid-basic, and ion homeostasis of the extracellular fluid, together with the redox potential. The next slide, shows the second cellular component, including the reticuloendothelial system. The cells found here are, among others, adult fibroblasts. They are number 1, embryonic mesenchyme cells, or totipotent fibroblasts, depicted here as by number 2, macrophages, number 3, fat cells, number 4, lymphocytes, here number 5, and mast cells, which are number 6. The totipotent fibroblasts are of major importance, since they in principle are able to differentiate into the various cell types of the loose connective tissue. Furthermore, adult fibroblasts form the extracellular matrix, composed mainly of collagen, which is C. Is it on this slide? I don't know. Oh, we will see that on the next slide. So, collagen, fibronectin, and elastic fibers. Together with the extracellular fluid, these elements are forming the extracellular environment surrounding and influencing every organ cell. The last component, consists of the neural component. The autonomous nervous system, parasympathetic and sympathetic, innervates the various types of via various types of ganglion cells, the loose connective tissue, as a fine network. In the previous presentation, it has been explained to you in a very thorough way. This innervation is most likely not in direct contact with the organ cells, and that has been said also before. It has been postulated that the neurotransmitters are secreted into the extracellular fluid, affecting both the organ cells and the cells of the loose connective tissue. By this system, cells of the loose connective tissue seems to be controlled by the autonomic nervous system, while on the other hand, it seems conceivable that the loose connective tissue can influence the functioning of the autonomic nervous system. Of course, here we are touching the zones of head which were mentioned yesterday by Dr. Beck, and he also showed us the consequences for thermography. So in summary, it seems to be this system surrounding all organ cells which is the prime factor determining and regulating the extracellular environment, or milieu interne. It is the connecting element or communication channel between cells and tissue, maintaining the interactions defining life. 4. The characteristics of a living organism is not that the various composing elements, cells, tissues, function, but rather that they function in interaction with each other. Recently, this system of ground regulation has been renamed the basic bioregulation system by Van Wijk and co-workers at my laboratory, including the possibility of influences of electric, magnetic, and electromagnetic fields, and possibly sound, on biological organism at this level. In this respect, it seems worthwhile to measure and interpret effects of external stimuli, like the various physical fields or acupuncture, as reported by several of the present speakers. Depicted on the next slide, it's at this level of the basic bioregulation system that we find the meeting of two streams of information. First, the influence of cellular processes on the physiology of the body, and via the body on its environment. So this information stream coming from here to there. And second, the influence of the environment toward the cellular level. That's just the opposite. Including the possible effects of the electric, magnetic, and electromagnetic fields. However, given the remarkable complexity of this system and its interactions with the organs, it's perhaps no wonder that still many questions has to be solved. One approach to solve part of the puzzle is to choose a single level, to attempt to analyze it in detail, and then relate the analysis to other levels crucial for the function of the complete system. Next slide shows in a very schematic way the levels at which we concentrate our research, or at least we have to concentrate our research. These levels include, here at number 1, the effects of chemical and physical environmental factors on the basic bioregulation system. This study will treat this system as a whole, as a complete system, as some kind of black box. 2, the fundamental biological aspects of the basic bioregulation system itself, including its regulatory aspects and the effects on the environment on its various elements. Third, the interaction between organ cells and the basic bioregulation system, emphasizing the basic bioregulation system as the environmental factor of the organ cells. And finally, at level number 4, the basic biology of the organ cells, including their mutual interaction, so the interaction between organ cells and organ cells. At present, research is going on at more or less every level here shown. So, for example, you have seen the research of Dr. Beck with the thermography of Dr. Stedbacher. In effect, they are doing their studies on this level. Also, at least that's my opinion, also the work of Dr. Smith is on this level. Dr. Ludwig, I think, will deal with this level too. Maybe Dr. Nordenström is dealing with this level here, number 3. However, what may ever be the case, here I will not treat all these levels, but instead concentrate on a topic which seems to be of relevance for all those levels. In the next part of this presentation, I will deal with the relationship between DNA as the genetic background of cells, cytoplasm, and the cellular environment. Fundamental knowledge about these relationships is of crucial importance to interpret the results collected from studies dealing with more complex systems. I will introduce to you and illustrate this topic on the basis of data collected within the current research program of my laboratory. This program contains, among others, research on processes involved in growth regulation and deregulation, the later resulting in dedifferentiated tumor cells. As pointed out on this slide, our starting point of our project can be briefly summarized by the consideration of the cell as a structure in a state of equilibrium between DNA, cytoplasm, and its environment. Of course, there's nothing new about this statement. However, it is less well appreciated that the existence of a particular cell is not confined to a single state of equilibrium. Different states of cellular equilibrium can be seen in processes as shown in this slide. These processes include the cell cycle, number 1, differentiation and dedifferentiation, number 2. Differentiation is number 2, and I pointed out here as number 3, the topic of dedifferentiation. First, one can observe differences between the early G1 phase and the late G1 phase of the cell cycle in terms of gene expression, metabolism, and responses to the environment. We will return to this a little bit later. Second, in the process of cellular differentiation, several discrete steps in coordinated gene expression and dependency on the environment can be recognized. The last process mentioned here, the process of dedifferentiation, will be treated here in more detail. However, already at this stage of my presentation, I should emphasize that this subject is not primarily treated from the scope of the cancer problem, but merely as an explicit example illustrating the idea of various forms of cellular existence. The next slide shows the characteristics of tumors originating from the same parental tissue. From the very early history of cellular pathology, pathologists observed diversity and morphological heterogeneity of neoplasms. Later, experimental oncology showed that in addition to the morphological heterogeneity, there also exists heterogeneity at the biochemical level, expressed in differences in quantitative enzyme levels and in the regulation of these enzymes. Most important, these differences in gene expression between the various tumors originating from the same parental tissue appeared to be non-random and coordinate or recognized. In the present discussion, it's of major importance that the various tumors, and again I am emphasizing it, originating from the same parental tissue, exhibited considerable biological stability, as was shown during continuous transplantation in animals for many years. These early observations indicate the existence of a multitude of aberrant but more or less stable intra- and intercellular organizations, which must be explained by the existence of various forms of tuning between DNA, cytoplasm, and cellular environment. In order to study this tuning in more detail, we are using at our laboratory a model system of normal liver cells, so-called hepatocytes, and several liver-derived hepatoma cell lines. Yep. This slide shows the various tumor cell lines and their degree of differentiation compared with hepatocytes. Of the forementioned cell lines, here we have the hepatocytes. We have two cell lines here and another two here. Of these 4 cell lines, we consider the cell lines MH1C1 and H35 as well differentiated, and the cell lines ATC and RLC as poorly differentiated. This characterization has been based on the difference in expression of liver-specific characteristics. As shown, the well-differentiated cell lines have retained, and we go into that in detail a little bit further, have retained the liver-specific characteristics to a great extent, whereas the poorly differentiated cell lines have lost those characteristics. So, by comparing normal adult hepatocytes and the well-differentiated and poorly differentiated tumor cell lines, we can make the following observations. First, and that's here, we can see a shift from the adult isozyme types towards the fetal type. The isozymes of aldolase and pyruvate kinase are well-known examples of this shift. Second, and that's here, yep. We can see a shift in carbohydrate metabolism. The expression of key enzymes related to gluconeogenesis decreased, whereas the expression of the key glycolytic enzymes increased with increasing dedifferentiation. Growth control here is altered depending on the degree of differentiation. For example, the poorly differentiated cell lines HTC and RLC are independent of external growth factors for continuous proliferation. More or less, they become independent of the environment. In contrast, the normal hepatocytes, in normal hepatocytes, well-defined conditions are needed for proliferation. These conditions include cellular contact, pH of the environment, and specific growth factors and hormones. In addition, alterations in the coherence of colony growth, the number of mitochondria per cell, and nitrogen metabolism, alterations in those processes can be observed depending again on the degree of differentiation. Next slide shows you how the morphology of cells is changing when the degree of differentiation is changing. Only look at the upper row. This is of no importance. Those here are H35 cells, and remember those things are well differentiated. Their cell morphology is more or less hexagonal, and the monolayer becomes completely closed. So they grow on a plastic substrate, and they completely fill up that substrate. Try to remember this picture, and then we're going to compare it with, only look at this one. We're going to compare it with the poorly differentiated cell lines. And here you see a complete different picture. The cells are not so well shaped anymore, and furthermore the monolayer is not completely filled. It will never fill. So here we see an example of a complete different morphology, and that we can show that this morphology depends on the degree of differentiation. As a matter of fact, the morphology of the H35 cells looks more like liver cells than the other cell lines. Most important, although not shown here quantitatively, these alterations appear to occur in a coordinate, non-random fashion. And I mean all those alterations, not only here the morphology. So for example, a decrease in key enzymes of the gluconeogenesis is paralleled by an increase of glycolysis and a decrease in the number of mitochondria per cell. In general, it can be said that the well-differentiated liver tumors are characterized by a high division potential, but with small alterations of gene expression, whereas the poorly differentiated tumors combine high division potential with large alterations of gene expression. These results show, first, that the heterogeneity observed for tumors grown in vivo can also be found in tumor cell lines growing in culture. And second, that the alterations of the various processes and characteristics happen to occur in a coordinated fashion. However, it must be realized, and it's only honest to say, that exceptions can be found, for example, at the level of expression of very specific enzymes, highly likely because of their own particular specific regulatory control. So using this system of normal culture liver cells and liver-derived tumor cell lines, we are able to study the various states in which a cell can exist. As mentioned before, phenotypical stability is one of the major characteristics of a tumor grown in vivo. We can also study this in our model system. Stability of the tumor cell lines in culture was studied by two approaches. First, heat production, and second, sensitivity toward external perturbation. Let's start with the heat production. According to Trencher, a cell has to maintain a heat gradient between the cell and its external world in order to maintain its structure or its existence, if you like. This gradient is reflected by the heat production of cells. So this slide shows you the heat production of hepatocytes, H35 and HTC and RLC. It's clear that the heat production of H35 cells is 2 to 3 times lower compared to the poorly differentiated cell lines and to the hepatocytes. These data indicate a smaller heat gradient between H35 cells and their environment, and suggest that H35 cells are less stable compared to normal cells, whereas the stability of the poorly differentiated cell lines is comparable to the normal cells, the normal hepatocytes. This suggestion was further investigated by measuring survival of the various cell lines after subjecting the cells to supranormal temperatures. In other words, to a perturbation of the external environment. The results show on the next slide. It shows that the survival of H35 cells is 17% after heat treatment at, for example, 44 degrees for 30 minutes, whereas survival of hepatocytes and HTC cells is approximately 50 and 80% respectively under identical conditions. These results show that in accordance with the results on heat production, H35 cells are unstable in a relative unstable state compared to hepatocytes and HTC cells. Furthermore, cellular heat sensitivity can be correlated with thermal sensitivity of the chromatin structure, and now we're going up to the DNA. Chromatin consists of supercoiled DNA together with many different proteins, namely histones and non-histone proteins. In the context of another line of our research program, consisting of the regulation of the cell cycle, we demonstrated, as shown in this slide, that cells in early G1, that's here just after mitosis, are highly sensitive compared to late G1 cells, which is something like here, Survival of H35 cells in the early G1 phase is 1 to 2% after heat treatment of 42.5 degrees for 1 hour, whereas about 10%, 10 times higher, of the late G1 cells, phase cells, survive this condition. Recently, we show that these differences in heat sensitivity can be correlated with differences in vitro phosphorylation of the non-histone chromosomal protein fraction of chromatin. So the degree of in vitro phosphorylation of non-histone chromosomal proteins, here separated by gel electrophoresis, increases dramatically in heat-sensitive early G1 cells. Here is the 37-degree level, and there if we treat the cells with heat. No such alterations were observed for the less heat-sensitive late G1 cells, which is shown in the right panel. It's commonly accepted that the capacity of in vitro phosphorylation of the non-histone chromosomal proteins is a measure of the chromatin structure. From these results, it can then be concluded that cellular heat sensitivity or stability is paralleled by the heat sensitivity or stability of the chromatin structure, indicating that the various states in which a cell can exist is paralleled by the various states of the chromatin structure. As mentioned, the cellular environment is considered as a major factor determining the actual cellular state, third factor. Also in this respect, no, I'm gonna skip this, I think. Oh, let's do it. Also in this respect, experiments on heat treatment of cells are of great value. Studies in which the cells were subject to temperatures of 41 or 42 degrees, comparable to temperatures of fever or local inflammatory conditions, revealed that in response to the temperature increase, the cells shift to a new, well-defined state. This is reflected by the de novo synthesis of so-called heat shock proteins as shown in this slide. Here the pattern of specific proteins synthesized, imaged by two-dimensional gel electrophoresis of control cells, 37-degree cells, that's the upper panel here, is compared with that after treatments for 30 minutes, and that's panel B and C and so on. It's clear that changing the thermal environment induces expressions of protein not or hardly expressed under control conditions. And those things are indicated by the arrows here, various arrows here. There's no proteins, and if you're going to compare that with here and look at the arrows, there you see new proteins which are not expressed in the 37-degree condition. Actually, this here represents the same at a different temperature. This expression of the so-called heat shock proteins is a very illustrative example of a site-specific gene expression by a site-unspecific physical factor. Another example comes from studies on salivary gland chromosomes of the fruit fly of Drosophila. Here, certain conditions and/or combinations of ions selectively stimulate the puffing pattern in isolated polyethylene chromosomes. This kind of control is highly unimaginable with direct action on the genes. Currently, this type of results are explained by the recognition that three-dimensional chromatin organization is the ultimate control system for gene expression. DNA carries the information required for its actual conformation in 3 dimensions by the sequence of its building blocks, the nucleotides. However, the non-histone chromosomal proteins play a crucial role in the final actual configuration. As we've seen before, the phosphorylation of these proteins is highly important for the configuration of chromatin. By this type of molecular alterations, the three-dimensional chromatin structure is influenced, resulting in alterations of gene expression. So again, these results indicate a state of equilibrium between the environment and the chromatin configuration coupled with specific gene expression pattern. Another factor making up the environment I would like to introduce to you now, and because of the time, I think I'll go up to my work with the physical vectors. Since the relation between physics and medicine is a topic of this symposium, and the fact that unfortunately Dr. Pop was not able to be here, I will treat the existence of optical photon emission from cells in a slightly more detail, although not theoretically, but rather experimentally. Based on two lines of evidence, it has been postulated that photons can act as physical vesicles for transmission of information between cells. First, some evidence have been presented that organisms, tissues, and cells respond to weak, non-thermic, and non-ionizing electromagnetic fields, ranging from the visible, far-red, microwave, radio, and extremely low-frequency portions of the electromagnetic spectrum. Various of the speakers of this symposium will show you impressive examples of this subject. With respect to the supposed interaction between biological objects and visible light, I will not deal with the well-known phenomena of photosynthesis, fission processes, and bioluminescence. Okay, on the next slide, yes, you see the second piece of evidence that there is something going on with light. This piece of evidence consists of numerous reports showing emission of optical photons from a large number of different species and tissues. The emission covers a wide range of wavelengths, ranging from approximately 800 to 200 nanometer. So these two lines of evidence have been converged to the idea that cells utilize optical radiation as a communication device with, in principle, the possibility of a very high information density and high speed. However, to be honest, it must be emphasized that this interpretation of weak luminescence by cells is still highly controversial and not generally accepted. On the contrary, most investigators in this field of research consider light emission of cells as an accidental byproduct of reactions which happen to be chemiluminescent, such as the generation of oxygen intermediates and excited carbonyl compounds, as also pointed out on this slide. In plant systems, a transition between at least two states of photon emission has been observed by Pop and co-workers. The first state represents the emission of plant seedlings in the presence of external light, experimentally determined immediately after the transfer to the dark chamber of the photomultiplier. During prolonged stay in the dark, photon emission decreased until a second steady state is reached. The transition between these various states has been studied in great detail by way of its decay kinetics, its dependence on the initial light conditions, and its spectral changes. In a sense, the technique of pulse illumination has been used, described as light-induced photon emission. The recognition of several states of photon emission stimulated us to study the decay of induced light emission in our model system of hepatocytes and hepatoma cells. This light-induced emission was studied in relation to the cell density. That's the other parameter we are studying. The next slide shows decay curves plotted in a double logarithmic way of the three cell types at low cell density, that's the left panel, and at high cell density, that's the right panel, after pre-illumination with monochromatic light for 3 minutes. The x-axis is time in seconds. It's clear that after pre-illumination of cells at high cell density, the decay curves of the various cell types differ significantly. The next slide represents a more detailed analysis of decay curves. Related to the cell density, panel A shows the maximum rate of emission immediately after shutting off the light source, the external light source, while panel B shows the rate of decay under these conditions of cell density. Comparison of the H35 and HTC cells shows that with increasing cell density, both the initial rate and the rate of decay induced light emission increased more rapidly in the poorly differentiated HTC cells compared to the well-differentiated H35 cells. Normal hepatocytes exhibit a complex cell density dependence. With increased cell density, an initial increase in both parameters was observed, followed by a decrease of emission probability. Panel C is of no importance right now. Well, I'll skip right now the explanation of this complex decay kinetics and cell density dependence, and I'll go to the end of my presentation. We will go into all those things a little bit further later this afternoon when we have our workshop, and I'll like to return to our original starting point. I showed you that a cell can exist in different but well-tuned states of equilibrium between DNA, cytoplasm, and environment. Or in other words, a cell can have different phases, so to say. This phenomenon was illustrated by comparing tumor cells with their normal counterparts. However, it was not the intention of this example to state that these alterations in the state of equilibrium are only confined to the process of malignant transformation. This example was chosen because of clarity. On the contrary, this phenomenon can be observed as normal, even necessary process. So alteration of the state of equilibrium occurs during processes of embryonic development and progression through the cell cycle. Extending this concept, it seems conceivable to describe other processes of illness, other than cancer, as shifts in the equilibrium between DNA, cytoplasm, and environment. Especially important in relation to what we have said about the philosophy of holistic medicine, such concept of cell stresses the importance of the interactions between cells and their environment as a major component in the process of illness. It shows that the regulation of the cellular environment by what we have called the basic bioregulation system can play a crucial role in various illnesses or sensitiveness toward the environment. A malfunction of this latter system can have profound influences on the functioning of organ cells, and reminding you the highly complex network of interactions between the various parts of the body on the whole organism. And those shifts, to me, looks very likely, as the last speaker of yesterday showed you, those quantum jumps in existence of a patient. it looks like also there is a sudden shift towards another state of equilibrium. With this, I like to thank you.